So I was thinking about this over lunch, and as I've said earlier, I'm not a soil scientist. I don't have an agronomy background. So I have a question for some of you, and I think, Bob, you mentioned this earlier. If you had to take a normal cotton production system and you were going to add so much nitrogen right to it, how much nitrogen would that be per hectare per year or per acre per year in weight? How much nitrogen would that be? No. Well, let's just say we'll pick a number. You know.
2 bales.
Okay. So, but when what I'm trying to figure out, and you'll see why. So, and again, I don't deal with that end of stuff. So, on a hectare or an acre basis, what's the weight of nitrogen you have to add to get 2 bales? What's the weight of nitrogen that you have to add in your fertilizer?
80 to 100 pounds.
So 80 to 100. So.
Well, you're talking units, right? You're talking weight of material.
Well, no, I'm not talking weight of materials. In other words, I'm actually talking about the physical weight of the nitrogen that gets added to that acre in order to produce that per year. What's that actual weight? 64 pounds? Yeah. Okay. And there's a reason for that. Okay, and I'm glad you said it's 64 pounds. Good. And I was hoping it was about that. And that's okay. I mean, it could be 50, it wouldn't matter. How many of you know, and it's something RM and I was talking to him yesterday, how many of you have heard or been involved in what's called the National Atmospheric Deposition Program? Good. Again, most people wouldn't know about it. This was something that was started in about 2000 and continued up until about maybe 2018, 2019. The U.S. Geological Service ran it across the country. And what they did is they wanted to figure out how much nitrogen simply came into landscapes across the United States from the atmosphere. Well, and this deposition was attributed to human impact. Our burning of fossil fuels, how much was coming into the landscape. And so what they did is they had stations around the country, like about 200 stations, and they collected it as wet and dry. I'm going to show you wet from 2002 to about 2017 for both ammonium and nitrogen. And the reason that I wanted to know that is I want to show you how much additional nitrogen is actually coming into our systems that you don't know about. And that's part of the reason things get screwed up, is because the atmosphere, through our activity, is putting more nitrogen in than what you know. In some cases, it may be 10% of the total nitrogen you add per hectare per year. So let me see if these are PowerPoints, and they should play. Let me go ahead and pull. So one is ammonium and one is nitrate. And I'm just going to start them, and they're going to be individual. So I'll have to give a little preamble. So on this slide, the darker the color, the more. In this case, it's over 20 kilograms of nitrogen per hectare per year is simply coming out of the atmosphere onto the landscape. 20 kilograms of nitrogen per hectare per year. So let me go ahead and start. This is ammonium. This is strictly ammonia. And watch where we are in Texas. You'll notice we're getting an additional 8 kilograms per hectare per year. But look at the Northeast. So they eventually do get it down. And so that's ammonium. Where all this is coming from, anybody know? Coal-fired power plants. And what it did, now, so if you look at Texas, my data that I've shown is the Lubbock region is getting an additional 2 kilograms, has been 2 kilograms of nitrogen per hectare per year across everything. That's the average. So you're adding 64, whatever, per hectare per year. You're getting an additional 2 kilograms of nitrogen per hectare per year of ammonia across that landscape. Now, in the Northeast, what it did is it acidified everything. It caused, that's what acid rain is. It caused decrease in streams and rivers and in lakes and killed off a lot of stuff. In the Great Smoky Mountains National Park, the amount of nitrogen and the acidification was so high, it killed forests. But I also worked at Big Bend. And Big Bend is the most isolated national park in the lower 48. It still gets an additional almost 2 kilograms of nitrogen per year. And you know what it did? It changed everything in the park. Over a 20-year period that I worked there, everything changed. All the soils never functioned the same. They started getting in invasive plants that they never got in. Because why? There was more nitrogen for those plants to use. So that's ammonia. What do you see what happens with nitrate?
That one said nitrate.
What's that?
That one said nitrate.
Oh, did it? Okay.
Yeah, yeah.
Well, let me pull up the other one. Ah, here's ammonium. Okay. So look what Lubbock and the region looks like in 2016. The amount of ammonium that was coming in just from atmospheric deposition. According to the map, we're somewhere between 4 and 6 additional kilograms of ammonium nitrogen per year, on top of everything else that's been applied. And we wonder why we have problems. Like I said, in Big Bend, it was there, at most we measured at all. And I was using those ion exchange resins. You know what we were doing? We were putting out funnels with resin beads under them. We were collecting the rainwater, then seeing how much nitrogen was coming in from the rainwater. And even though it rained maybe 4 times a year, in some cases it was like 2 or 3 kilograms of extra nitrogen per hectare per year because of the human input of nitrogen in the atmosphere from the burning of fossil fuels. And it's that dynamic that, again, I was talking about mindfulness, that's what we don't pay attention to. We're contributing to the nitrification of our planet as a consequence of simply burning coal. And that's what's going on top of all the stuff we have been planting, we have been using. Sir?
What's the explanation why the nitrate went down and ammonia went up?
Part of it has to do with the atmospheric chemistry in terms of what's being burned and how it's being distributed. Yeah, and again, I don't know enough about the chemistry to know why ammonium is so more than the nitrate level. A lot of it has to do with the rainfall and the chemistry of that rainfall. So let me ask you a question. What's the pH of vinegar? What's the pH of vinegar? It's about 3. We measured the pH of rainfall in Big Bend, 3.2. Yeah, it was raining vinegar in Big Bend for a couple years. And you're talking about Big Bend National Park. It's in the middle of nowhere. I don't know what the pH of Lubbock was like at that point. It's probably about 4. right? You know, it's a good vinaigrette in terms of pH. But those are sort of the unknown factors that occur. And again, you know what happened when so if you look at the middle of the country and the Northeast, what happened up there is it changed all the dynamics of the forest and croplands in terms of decomposition and everything else. Because the fungi are dealing with all this other nitrogen. We're fertilizing and all this other nitrogen is coming on top of it. Now, nitrogen always is going to come in through rainwater. Lightning makes some of it. But we put a lot up just in the burning. It's one of the reasons to get off coal. We've acidified too much. We put too much nitrate and ammonium up in terms of that. Right? So the reason I want to talk about that is I want to get into this whole question next of decomposition and enzymes and sort of work through that. Because if we're trying to understand how you actually build soil carbon and what happens to your stubble and root material, all that plays into that. Right? Okay, so that's where we're going to go.
Dr. Zach, why does that not follow a rainfall pattern that you would normally expect to see? Is it just that, like in Big Bend where it hardly ever rains at all, that the rainfall itself is capturing more of the nitrogen?
No, because so it turns out that this was wet deposition, but a lot of it comes in as dry deposition too. And so in this particular case, even though they're just measuring what comes in as wet, when it rains over that year, it's still depositing that amount of nitrogen. It's just not going to be spread out. It's going to come in as a slug. So when you equalize it across the year, it's that amount per year. Yeah. So let me ask you folks a question, because this raises something that come out of Big Bend. Can the amount of nitrogen in your soil in the form of nitrate ever be toxic? Hell yeah, right? You get up to certain levels. So those of you that are crop consultants, and again, I'm working the ecology component. When you take the numbers out of agriculture, we don't use the same scale. So there's a measurement issue. So can you give me in parts per million nitrate in your soil? What would be more than what you would want? Anybody have a number? 120, 130?
40 to 50.
Okay. Let me tell you about something at Big Bend.
For a crop.
Yeah, for a crop. Okay, and that's probably true of everything, Bob. Yeah, 30 to 40 parts per million. We had, if you ever, how many people have been to Big Bend? Okay, so when you're coming down from Marathon, so when you're coming from Marathon on the main entry into the park, you go through the check-in station up over the hill and down to those flats, and there's a lot of clay. There's a lot of degraded grasslands down there. I think the first major hiking area is Dog Canyon. You come down and see Dog Canyon. We worked on that for a while. And they wanted to try to revegetate. It was a Tobosa grassland. Now, what was interesting, and now you think this is Big Bend National Park. We were interested in finding out why you had these bands of vegetation. Why did you see bare vegetated? And a lot of it had to do with collection of moisture and whatnot. And then we looked at it, we said, okay, there must be some microbial component to it. So what we did is we said, okay, we're going to sample the soil and look at microbial biomass, carbon, the fungi, the bacteria, and look at the nitrogen content. And what we discovered is that in the top 15 centimeters of soil, whether you went from a bare area to a vegetated area, it was relatively about the same. And we said, well, that doesn't make any sense. What's accounting for the stripes and the pattern? We said there has to be something else going on. So we started digging below 15 centimeters. So at the surface, the nitrate levels were 10 parts per million. You went down to 15 centimeters, they were 80. Go a little more, they were 120. By the time we get down to 45 centimeters in depth, the nitrate levels below those grasslands, where it was bare, was 450 parts per million of nitrate. Now, if you looked in the areas that were vegetated, it never changed with depth because there wasn't any vegetation to use it. And so what was happening was, what we postulated, and was probably true, is that the deposition was coming in year after year as this atmospheric deposition. There were no plants to use it, and the microbial component couldn't respond because there were no plants. And so the reason there was 450 parts per million deep down is that was the depth at which the water, when it rained, actually went. And it just kept moving all that nitrate, nitrogen to that depth. 450 parts per million. Right? Talk about, and that's what happens in these systems, or any system, year after year after year. So the problem was, you could not revegetate that system until you dealt with the excess nitrate. Because every time they plant grass in here, it die. Well, yeah, it's going to die. The nitrate levels were far too high. So what we ended up doing was changing the temperature dynamics on these systems by simply putting brush. Once you got the microbes to start using and building biomass again, the nitrate levels drop and the grass grew. But you had to deal with that, but you had to deal with the nitrate. And again, where was that nitrate coming from? They didn't fertilize. It was all coming in after year after year after year after year, last 40, 50 years, 60 years of atmospheric deposition of what? 1.2 kilograms of nitrogen per hectare per year. And that was enough to tip the balance on the system. So, I mean, again, now, they've stopped they stopped that program completely. They don't fund it anymore. So we don't know what the dynamic is anymore. But you have to keep that in mind as we look at how we fertilize, because the atmosphere and us in general is over-fertilizing the planet as a consequence of fossil fuels. Irrespective of climate change, we still put up too much nitrogen in the atmosphere. That gets washed out as ammonium and nitrate. Now, this is all going to come into play when we start talking about decomposition. So, what is decomposition in terms of its biological? It's simply the conversion of complex carbon into simpler forms that get used up by microbes. And there's two parts to decomposition, right? There's an abiotic part in which the complex materials is broken down to simpler forms, and that's by UV degradation. But one of the reasons why we lose organic matter on our soil surface has nothing to do with microbes. It simply has to do with UV breakdown of chemical bonds because of the amount of UV we get here. That's going to happen no matter what. I mean, think about how the color of your car changes inside over the years as the UV hits it, right? That's bond breakage. And then the other part is that component, which is biotic decomposition, which is carried out by the microbes. Now, the reason I spent so much time this morning talking about how fungi grow, here's a crucial thing. Bacteria can only grow on the surfaces of materials because they're relegated to a water film. But fungi, because of the hyphal tips, can actually penetrate material. They exert enough pressure that those tips can grow into anything into that material. And so what fungi do with a volume is not only grow over the surface, they grow into it. So fungi are decomposing material and using material not just from the surface down, but to inside it out. And so they decompose it in both dimensions. But bacteria can't do that. They're relegated to that surface. So in terms of what the decomposition process is on the planet, it's almost all fungal driven in some way, shape, or form. I mean, obviously, we do have this abiotic decomposition. That accounts for all the microplastics in the ocean. It's why a plastic bag in your yard, if it sits out long enough, is going to fragment all up. It's not decomposing, it's just breaking down to smaller pieces. But that's because the bonds that hold the plastic together are breaking down. So again, fungi are responsible for 70 to 95% of all decomposition. That's what runs the planet. And the reason that this occurs is fungi do not need a complete water film to grow. As long as some part of that mycelial network has access to water, they translocate it in and grow across that surface. Why? Because they have a cell wall that moves them along. They're protected from desiccation. And they can extend them over long distances. Now, where this comes into play for today is that they produce a large amount of enzymes, but fungal enzymes are actually excreted into the environment. They're not internally like ours. They excrete them into the environment in which they work. And if there's water, they work more. But once a fungus excretes enzymes into the environment to break down carbon, that enzyme, as long as it's not degraded, functions even if the fungus is not there. Why? Because it's been excreted. It's like it's like acid. It'll just keep working on it and working on it and working on it as long as conditions are favorable. So fungi produce what we call exoenzymes. Now, I may have put this up one time before. The world in terms of carbon is very diffuse in terms of what's available as carbon. So if we look at some of the carbon that's very simple, we go from methane to alcohols to sugars to disaccharides, monosaccharides, starch, right? A lot of fungi will use these because they're easy to use. But as you get to this right side of the curve, starch, hemicellulose, cellulose, lignins, chitin, keratin, lignin. These are the components that make up microbes and they make up plants. It's this side of this curve that constitutes most of the carbon that fungi deal with in the environment, because it's all produced by microbes in plants. So what controls decomposition rates? Well, it's the carbon-nitrogen ratio of the material, the type of carbon, temperature, moisture, nitrogen levels, and the rate of grazing by the microfauna. All these come into play that regulate how fast that decomposition occurs. And so when all of these are optimum, that carbon is going to be used because fungi are using it to grow and build biomass. So these exoenzymes, right? And in the case of fungi, they can either be induced to be produced or they can be repressed. It depends upon how the fungus in the environment is actually going after that carbon. And as I said here, zygomycetes such as Rhizopus can grow on substrates in which they don't have the necessary enzymes to use. Will a zygomycete grow on paper by itself? And the answer is no. Will they grow on paper when something else is growing on it? And the answer is yes, because they use the breakdown products of those enzymes that get excreted. And again, this is why they're in your fields. And that's why we call them secondary sugar fungi. They don't carry out the decomposition themselves, but they're pretty good at taking that simpler carbon away from the other fungi. Now, something else you need to know. I spent a lot of time talking about tips because the fungi grow at their tip, but this is the region where they're also producing a lot of the enzymes that they use for growth, because they don't do it along the full length of the mycelium. They only do it where the resources are and those tips are actively growing. And then from those tips, they also produce antibiotic and other products to keep other organisms away. But that's the reason why as fungi grow across something, they may start out as simple threads, and then they'll produce this very diffuse kind of structure that allows them to take up those nutrients in a much more efficient manner. Now, I think it was either Gary Zimmer or Dr. LaSalle showed this somewhat yesterday, a different version. The carbon-nitrogen ratio is critical because what this indicates is how much nitrogen do you need for the organism to use on a given amount of carbon. And so bacteria on this figure are 3 to 1 to 5 to 1. So what that means is for every 3 to 5 grams of carbon that they use to build biomass themselves, they need a gram of nitrogen. Fungi are more efficient. They do 15 to 1 to 20 to 1, right? So for every 20 grams of carbon they take in, they only need a gram of nitrogen. And then we as humans are 25 to 1. And then wheat straw itself as a product is 80 to 1, and wood is somewhere between 200 to 400 to 1. Now, the reason that that's important is that the bacteria cannot grow on this stuff. They'll start, but they won't grow. Why? Because there's insufficient nitrogen in the material to actually use it, unless you give it to them as fertilizer. And so we all do it, when we fertilize anything, we lower the carbon-nitrogen ratio, technically, of that plant material to allow the organisms to use it. And the bacteria specifically. Because bacteria are so quick in their growth, they also have, a high nitrogen demand. Whatever nitrogen you add that is not immediately taken up by the plant, they will use it to use the carbon. And they will sequester it. And they will consume it. And that's why we keep saying you don't want bacterial dominated fields. They use too much nitrogen. You want a field that's more fungal dominated. Plus also, the bacteria can't decompose the way they do. They'll use whatever is there, but they won't build the necessary components to build soil organic matter. Now, why are fungi able to use wood? And that's because they're able to translocate nitrogen from someplace else so that as they're working on the carbon, they essentially bring that carbon-nitrogen ratio down to 15 to 20 to 1. I say this all the time. Besides the pizza, lock the doors, put in some porta potties and some water. There is enough carbon in this room to keep us alive for a week. It's more than enough carbon. There's a lot of calories in here. What's not here is the nitrogen, right? Technically, this carbon is available for us to use. Now, we don't have the enzymes to break it, but if we could, we need the nitrogen. So your diet is based on a 25 to 1 ratio of nitrogen to carbon. We have to have that, otherwise we will die. And so what's happening in your fields is the organisms, the bacteria and the fungi, are always looking for ways to get that carbon-nitrogen ratio down to what they need to use the carbon available. For a bacterium, if you're giving them more nitrogen, they're going to consume as much carbon as they can. The fungi are going to do that too, but they do other things with it that bacteria can't. Now, if you look at a plant's carbon chemistry, in terms of what's available, simple sugars, proteins, it's the cellulose, hemicellulose, and the component called lignin, which provides the majority of the carbon that fungi have to degrade. And that's true of almost most plant parts. I mean, as you well know, as Arnon was saying this morning, the fibers off of the cottonseed are all cellulose. The stalks and stems and some of the root have lignin in it, right? So that's a much more recalcitrant carbon source, but almost the majority of it is cellulose in some way, shape, or form. That's why they want to use corn starter and other things for biofuels, because what are they doing? They're converting that cellulose, or they're converting the complex cellulose into glucose to make what? Alcohol. That's how they do it, right? They got one group of fungi to convert the cellulose to glucose, and then they get a yeast to make the alcohol out of it. And that's what we burn, right? That's what we burn. So again, just a little bit of what structural components are necessary. The calculation, 700 billion tons of cellulose on the planet. And again, where's the cellulose? It's in the cell walls of the plant or in the cotton fibers. And again, just a different version. But cellulose is just a polymer of glucose. That's all it is. It's just a sugar, a monosaccharide strung end to end to end to end. Now the problem is, how do you break these bonds and release the glucose molecule that then goes into solution that the microbes then can use? And that's a different story. So it turns out that majority of cellulose decomposition is by fungi, and there are two enzymes that they excrete. They excrete an endocellulase, right? And what it does is it cleaves these bonds between the sugars within the molecule. It simply goes in there and chops them up randomly. And then you have what are called exocellulases, which break off two glucose molecules or random sizes, but they do it from the outside. So one set of enzymes goes in and cleaves this molecule up anywhere. The other one starts at the end and starts cutting it in groups of twos. And by doing that, these smaller molecules are then made available for the fungi to take up and grow. And that's how they produce their biomass. But you need both. You can't have one or the other. The systems to use cellulose is designed that there's a synergy between the endocellulases and the exocellulases. You have to have both in order to do it. And again, just a cartoon version of what's going on. So and this one they call glucanases, that's a technical term. There we go. So you have one that chops them up in between, and then you have the exoglucanase, which is cutting off these two units of cellobiose, and then are broken down by another molecule. And this sugar then is actually in solution, right? This molecule is dissolved in water in the same way that you put sucrose into your coffee and dissolve it. It's in water, and then the microbes are able to take that molecule up and bring it into their cells. But all cellulose is degraded like that. But you got to have the fungi that produce those enzymes, exocellulase and an endocellulase. So what's happened over time, right? So one of the problems with here, as I've mentioned, is that fertilization brings the CN ratios down so that the organisms can use the carbon that's there. And at the same time, in here, as the winters in the Southern High Plains get warmer, optimum temperatures are going to allow them to do that more. And then what do you do by fertilization? You increase the decomposition. And that's not always a bad thing, but there's a tipping point where it becomes too excessive, and then you can't keep carbon in your soil. Because as long as it's warm, and as long as there's moisture, the microbes are going to grow, and they're going to use that carbon. Now, the other part of the story is lignin. And lignin as a molecule is a really complex biopolymer that covers the cellulosic walls primarily in the vascular tissue, right? But what's interesting is it's only basidiomycetes and some ascomycetes that have the capacity to degrade lignin. The ascomycetes and basidiomycetes can degrade cellulose. Some bacteria do. They work, they're soil bacteria. Some do. But what we've discovered over the last 15 years is that nitrogen levels in your soils or coming in from the atmosphere suppresses lignin degradation. It'll stop it. And the reason is that excess nitrogen influences the ability of the organisms to produce the enzymes that are necessary to degrade lignin. And then in these systems where you have lignin, that stops, but you don't get the byproducts of this to build soil organic matter or to build fungal biomass. And this is what's happened across the planet with the atmospheric deposition. We have changed how lignocellulosic material around the world has changed as a consequence of deposition. Now, what does this thing look like? If those of you like chemistry, it's a phenol propanoid unit. So if you know anything about phenol, here's the ring structure. It is a very stable ring. It's very difficult to break those bonds. And it has different units to it and different kinds of alcohols that make it up. But this is the molecule. This is why wood is wood. This is what gives wood its tensile strength, its strength to it. It's encrusting those xylem vessels in wood. But the fungi have to degrade this. And there are two ways to degrade it. There's what's called orthofission, where you break the ring in this manner, or you break the ring in that manner, metafission. And so fungi, mainly the basidiomycetes, are the ones that produce the enzymes to do that. Right? And so, again, little technical terms. The major enzymes are lactases, which initiate ring cleavage. Right? Now, what's interesting is some bacteria have the capacity to degrade pesticides. They have an extra bit of genetic material called plasmids, but they will not degrade lignin. Bacteria can't degrade lignin. So it's the fungi that are involved in that. And then they also produce these enzymes called lignin peroxidases and manganese peroxidases, which then destabilize the ring. So once they're able to destabilize the ring, they start working on all the rest of the lignin and the cellulose to start decomposing. And I got this out of somebody was looking at this in terms of cotton stalks for alcohol production for ethanol and alternatives of fuels, right? You know, the amount of cellulose in wheat stalk is 32 to 46%, hemicellulose is 20 to 48, in close association with lignin. So, you know, the stalks that are left over after harvesting this lignocellulosic material plays an important role in why the systems are able to function. Even grasses have some lignin in them. They don't have a lot, but they have some. And I mention this because of the fact that, you know, this whole question about bacteria, people say, well, don't bacteria carry it out? And their answer is, no, they have some of the same enzymes, but they will not degrade lignin. Now, what kind of actual decomposition of lignin is there, right? And so there are three basic types. And again, ascomycetes and basidiomycetes are the ones that do this. And I would tell you, a lot of this information is based on wood, but it does occur on any kind of lignocellulosic material. So you have what are called soft rots. And these are ascomycetes and deuteromycetes. They're anamorphs. And really what it is they degrade the lignin and hemicellulose, and they put cavities in the material. So that's why it's called a soft rot. The material is there, but it's where your deck or your fence material or some other that's lignocellulosic feels soft to the touch, right? And again, occurs in woody materials that are moist and have adequate nitrogen, right? And this is least specialized by the fungi. Ascomycetes do it. And again, is it going on in your field if you have cotton or corn? The answer is yes. It's just at a different scale. Now, there's also what's called brown rot. And this is where the wood takes on, the woody material takes on a sort of a crumbly nature. And in this particular case, it's sort of crazy. The fungi in a brown rot use the cellulose and then leave behind the lignin, which oxidizes and turns brown. And so most of the fungi that do this are basidiomycetes, and I should have put on here some ascomycetes. But what does that look like? Well, I'll tell you in a minute. And then we have white rots, in which these fungi are removing the lignin but leaving behind the cellulose. So some ascomycetes and many of the basidios that grow on woody material are like that. So what you find with lignin is that you actually have a complex group of fungi, ascomycetes and a lot of basidiomycetes, that do different things with it. And as a consequence of this, those breakdown products are what are contributing to soil texture and soil health. Because if you're growing on cellulose alone, they don't produce enough breakdown products. They will simply use it all and build biomass and respire the rest off. But it's the lignin components, the more recalcitrant material, that actually contributes to long-term carbon storage, because it's difficult to completely use. So for those of you who have not seen it, this is what a brown rotted material would look like. It's sort of cuboidal, really crumbly. Another example in this stump. And again, people show this a lot for wood, but this is the same thing that's going on with your cotton stalks. It's the same thing that's going on with your corn stalks, where there is that material in it. And this is what the ascomycete would look like. This is Deldinia. Or you have Tremades, Schizophyllum. Now, this is soft rot. Soft rot, like as I said, uses the lignin, or not soft rot, but right rot, uses the lignin and leaves behind the cellulose. So what it does is it produces a paper-like material. And different types of fungi. Now, could you see these out in your field? And the answer is probably not. But are they there? Yes, they are. Otherwise, you would always have cotton stalks. They would never decompose. Those of you that like shiitakes, you know, they grow on wood. So you could probably grow them on cotton stalks also. You know, if you wanted to farm your field for fungi, just use your cotton stalks for it. Some other kinds of white rots. Now, how do we measure decomposition, really? Well, a lot of it is just weight loss over time, some enzyme production, CO2 production, but this is probably one of the most important parts, is following how much mass is lost of that material. And there are 3 phases that occur in any kind of plant material over time. So the phase 1 is where you have the leaching of any solubles, microbes start to colonize. You have phase 2, which is your cellulose loss, and eventually you get down to your lignin, and this is what takes the time. This is what contributes to your long-term soil carbon storage. This is what you're going to build up in sequestration. It's the lignin material and associated products that are responsible for that, because it takes decades for this material to eventually be broken down. Now, I like this graph because it shows a number of things. What this was doing was looking at wheat straw, and what it's showing is, okay, over the same period of days, 80 days, how much biomass do you build up, how much total carbon remains, and how much of this is extractables, cellulose, and lignin over time. And so what you can see is most of the microbial production in any plant material decomposing is from the initial solubles and the cellulose. Once you start getting down to the lignin, you start to have fewer and fewer fungi that are able to use it. But again, I keep pointing out, it's this component, the C3 line, the lignin line, that you want. So why does decomposition lead to mass loss? Well, some of it is strictly fragmentation to smaller pieces. Some of it is respiration of CO2. And then here's where it all connects up with soil health. So you have your crop residue, you have the decomposition, your microbes, but from that decomposition and the microbes comes out this humic materials, which then is crucial for soil structure. It's this component here, as I'll show in a minute, that's what's responsible for your long-term storage. It's not the plant material that comes on initially, because it's being used by the organisms, but it's the breakdown products of the microbes and the plant material that are more difficult to decompose to produce these structural compounds. That's why your soil goes from orange to black. It's the humic materials that are in it that are the long-term carbon storage. That's what you're trying to build. That's what makes coffee coffee. It's the humic materials in it and the phenolic compounds. Okay. Any questions about this part? Yeah.
So, of course, I've never grown corn, but reading articles, you know, over the last couple of years, how they're having to deal with extra corn residue, corn on corn.
Right.
And one of the things I was reading was they were blaming it on GMO corn. The stalks were thicker, stronger. They were being bred to where they would stand better, and that was the problem of why they weren't breaking down quick enough. The solution was more tractors and tillage.
Sure.
But with the lack of rotation, I'm assuming it's also part of the biology is degraded to that point where...
Yeah, so the reason they're stronger and can stay up more is because they have more lignin in them. And so the lignin is only degraded by the basidiomycetes. So if you don't encourage that as part of that system, you're not going to get them to be... And lignin's going to take a while longer, right? Simply as the nature of that molecule. But again, it's those breakdown products specifically from lignin that contributes to the long-term health of that soil. I mean, the problem is, in natural systems, that wouldn't matter. It's just when you keep putting that amount of primary production on year after year after year, you run into the problem.
So if we need, so I'm guessing just in layman's terms, we need for a longer term carbon, do we need more woody stemmed plants in our cover crop rotations? Something with more lignin in it?
You do, but again, if you're going with a corn, cotton, even soybeans, right, or black-eyed peas, they have enough lignin in them to make it,
Okay.
right? Yeah, because
I'm either cotton or cover crops on my drive.
yeah, cotton has enough lignin that again, because of the stalks, it's going to produce enough lignaceous material to be able to contribute. Because you don't want a lot, like you said, if you have too much lignin, it's going to take years for it to break down, right? It simply will, right? Yeah, good question, Bob.
You know, it's kind of amusing, you know, you talk about GMO corn, you know, a lot of companies recommend spraying GMO corn with fungicides to improve standability.
Yeah.
And I've never seen any difference in physical standability between GMO corn and non-GMO corn in the field. You know, they're all worried about it falling down, whether or not it decomposes at a different rate. You know, I don't know.
Right, that's right. And that's something, I mean, we do know that material that's been treated in different ways has a hard time decomposing. So I had a friend of mine, this is good, I had a friend of mine that worked for the Department of Defense. He was a soil ecologist. And they hired him because what the Department of Defense wanted to do was to set up a sustainability program where they would shred documents and compost them. Nice, just cellulose. And what he discovered was it never decomposed because they bought paper that was impregnated with some other kind of chemicals that kept the paper from being degraded when it was in the file cabinets, but if they got wet. Right? And so he couldn't compost them because the material was actually antimicrobial. And so you couldn't use it. So they had to stop doing that. Right? And it's the same thing. I mean, you know, once we start modifying what fungi and bacteria, I know I sound like a broken record, once you start modifying that, the capacity of the system to process the material that's on there is affected. And you either have too much trash that you can't deal with, or depending upon the spacing and amount. I mean, I just had a thought and it just occurred to me. If you do 80-inch cotton, will you have enough stock to actually contribute to soil organic matter formation? I don't know, right? Because you need a certain amount. And again, that's where all this comes into play. It's not one or the other. It's all of it taken together comes into play in terms of how the landscape, not your row, but the landscape is processing carbon to build what you want in that system. That's an interesting question. I don't know that. I don't know that answer yet. But it's something to think about. Is there a minimum amount of trash that has to be produced to be able to build soil organic matter around here, as we were talking about, right? Given that nature. And it might be good for productivity, it might be bad for soil health in the long run.
Well, that's interesting that you bring that up because I have noticed, especially on my irrigated ground, wherever we run the strip with the burr extractor, underneath the extractor chute where we're dumping all the burrs, which are some of the softer tissues, you know, some of the shorter limbs, it's coming out of the burr extractor falling on the ground. That is my best wheat.
Yep. Yeah, because you're putting that carbon back in for it to be processed. Yeah, yeah. Yep. Okay, anything else? Okay, I want to, let's see what time it is. It's 1:54. Where are we at here? Oh, good. I have a little bit. I want to pick this up in a very different way. What I want to talk about in a little more detail is how do you actually form soil organic matter? How do you actually form it? What are the compounds involved? So I have a little short aspect of that. Okay. You know, throughout today we've talked about fertilizers versus organic fertilizers. You know, I put this in there just to cause some discussion more than anything else. I'm not necessarily sure I believe it, right? But it talks about what do synthetic fertilizers do? Well, according to this diagram, they feed the plant with plant nutrients. What about organic fertilizers? Well, they have both the organic component, like the manures that in some ways that contribute, or the stubble that feed both the nutrients and the organic matter. So in a functional ecosystem, this is going to be what's going on. This will not at all. This is your atmospheric deposition route, the nitrogen that comes in from the atmosphere, right? Okay. So, what are our sources of soil carbon? Well, the plant litters the most. We get it from the microbial cells, microbial microfauna, animal waste, fur and hair. Anything that's in the environment that it's made out of carbon that's naturally made is going to go ahead and be processed in some way, shape, or form. The issue is this is the largest in any system. Right, and so you all know this about soil health and about cation exchange capacity, nutrients, influences roots, right, is our soil organic matter. Now, here's where it becomes increasingly more difficult to understand and to be able to evaluate because of the chemistry. So humus, we all know, is what gives soil its texture, its earthy color, its brown color, the humic materials, but they're breakdown products of both plant and microbial component. And it's the humic materials in soils that you're trying to build up for long-term soil organic matter formation. This is what you measure. You measure both the material that's there in coarse form, that gets incorporated in some way, shape, or form from earthworms and other things that are brought down. That's part of soil organic matter too, but that's the short term. The long term are these more complex humic materials where the majority of carbon is stored. That's why a mollisol is a mollisol. That dark structure of soil out at the top is all the humic materials that have been produced by the decomposition of that litter material that came on those grasslands. And even around here, you look underneath your stubble, you look underneath your leaves, you're going to have that humic materials that are generated. They have to be, because the complexity of the carbon compounds in that material. Now, here's what we know so far, and there's different ideas about this, right? One of the primary components that's involved is an amino acid called glycine that gets generated from the plant material. And then you have the lignin material that gets degraded into these phenolic aldehydes. Some get used by microbes, but you make these polyphenols, and then you have the cellulose and other non-lignin substances interacting with it, and you get these compounds which are humic acids. And so it's these interactions between the microbes and the plant breakdown products of decomposition, along with fungal cell walls, bacterial cell walls. Do you recognize this molecule? Melanin is what gives our skin color, right? Fungi produce melanin. It's a complex carbon compound, right? So it gets put out too. So these are all complex carbon compounds along with lignin. And, they produce a variety of products that end up being humic acid. This is a very more structurally significant component, and if you're interested more in the chemistry of how all this thing works. But start out with lignin and other plant phenolic compounds, right? Organic residues in general, and all this stuff contributes these breakdown products until you end up with, phenolic radicals and hydrobenzoin quinones, polymerization, and you end up with this humic acid material. And this is why your soils turn brown. This is what adds structure and builds porosity. This is what, because these humic materials have charges on them which change the structure of your soil. And once you get to this component, the only way to lose it is to burn it off. So if you have a fire which is hot, it's going to burn off humic materials in your soils, and you're going to lose all that. And how long will it take to get here? 20, 30, 40 years? 50 years? These are long-term processes, right? They're not something that's going to occur in a year. They're going to start, And they're always going to be going on. Unless, see again, that's the problem with breaking up the fungal network. Because if you break up the fungal network, you never get to this stage. You can add all the organic matter you want, but you never get to that stage.
So you're saying like if you have a grass fire, it's going to be hot enough that it's going to take out humic acid in your soil?
It depends upon how much litter's on your soil surface and how deep it penetrates. Yeah.
So probably like a fast-moving grass fire, no big deal.
No big deal.
But like a big old forest fire that's really getting hot.
Yeah, if you have a lot of material on your soil surface and it's going to burn hot for a while, it's actually going to burn and volatilize some of those humic materials in that top 5, 6, 7 inches. And that's the detriment of having, and then we know that for forest fires, that's the problem. Grassland fires, if they haven't burned in a while, that's the problem. They're gonna start volatilizing a lot of stuff that was going on. Stephen.
No, so along with that, looking at some CRP grounds, been grass for 20, 30 years. Gonna slowly bring it back into crop production. How would be the best way to, don't really want to plow, don't really want to do much tillage, but to bring it back into production, how do I deal with the residue that I do have left?
That's a good question. In the little bit that I've been involved with, one of the things we looked at was, and again, we don't do it. We wanted to find out before we even did anything, what was the rate of mineralization already occurring. So if we know how much nitrogen was moving through the system, we could then predict what we had to do. Because if we didn't get enough nitrogen without having to fertilize it like crazy, then it wouldn't matter about the residue, because we know the residue is decomposing at a rate fast enough to put that system back into equilibrium.
My best idea was just to mop grade, bring it down short,
And just plan into it. Yeah. But before I would even plan, I check what my mineralization rates were like, and what my pools of nitrogen are. Not just once, but several times over a year to see what they look like. Then you can get some idea of what has to happen. Yeah, then do that. That's a good question. Anything else?
Quick question.
Yeah, Nora.
You said there was some different ideas on this, and isn't it kind of a new thought that, humus is not made from humic acid, that those humic acids were from the extraction process in the lab?
There are, yeah, and I
Touch on that or not? Or if you don't want to.
no, I'm not. Yeah, I wouldn't touch, because I'm not a chemist on that level. What I would say is, and I've read that. It's a question of how we analyze this stuff. Because like you said, I've read reports that say, yeah, we get the humic acids only in the lab when we do all the extractions, right?
The guy
Yeah,
that wrote the soils textbook, they rewrote the book.
right.
What was his name? Y'all had him in no till.
Mm-hmm.
Yeah.
Yep. Yeah, and now.
I can tell you even Katie Lewis she's on my committee, and we don't talk about like humans, because we break it into different parts of the carbon
Cycle.
cycle.
Yeah.
Is it particulate carbon? Is it soil organic matter? So yeah, you're right. In the soil science world, we're even changing the conversation.
Sure. And again, part of it, so, and that's the interesting too, because if you look at it from the fungal ecology world, we don't talk about it that way, right? We don't. We talk about it from this perspective, because this is what the chemistry is for the fungi.
Do you feel like adding humic acid is worth anything?
I don't think so. I would just simply put organic material down. The problem is, you don't know how well what it's going to do when it's there, if it's going to make any difference, right? You have to test it and see, but I would rather just add some organic source, and then let the system process it.
Biochar.
Yeah. Yep. So again, in the world I come from, this is what we talk about in terms of what these things look like. Now, here's where it goes, the newer stuff. This is a 2020 conversation. And so, and it goes back to what has just been said, is How much of this is part of the fast cycle? This is the particular organic material, right? Yeah, and again, I mean, this is where we're looking at. So lifetime is 1 to 50 years, fast cycling, but this is important too because this is what's feeding the microbes. This is what's coming in from your roots and everything else. At the same time, this is what you have to build also. This is your slow carbon. 10 to 1,000 years, it's going to exist. And the question's always been, what are those small organic molecules? I mean, again, coming from the fungal world, it's all the same things I talked about, the humic acids, humates, the breakdown products of lignin, and the breakdown products of fungi, right, are contributing to it. This is probably the area we're still working on. We don't have a good idea of what constitutes this. All we know is, you want good soil, you don't only have to do this, you have to do this. You have to do both. And you have to allow both to occur. But we're talking 1 to 50, and we're talking 10 to 1,000 years. So, you know, in a decade, it's doable to start changing the system. It's just a question of how the plant material and the fungi are going to be involved in that. And I showed this before, you know, in terms of our understanding about what goes on in these compounds and slow carbon and passive carbon. So what we're looking at really is these are your dollar inputs to your system. The soluble compounds, cellulose and lignin coming in, they're being processed by the fungi, microbial plant products, slow carbon, passive carbon. All this is increasing over time, right, in your system in which you're not tilling, right? But soil temperatures, moisture variability are going to regulate all this. That's important. However, when you get into fertilization and tillage, what you do is you still have the same inputs, right? Still the carbon currency. But what happens is you notice that in this case, the biomass is particularly the bacteria. And what's happening is these bacteria are pulling carbon because of the carbon-nitrogen ratio out of these pools and simply respiring it off. They're not building any of this. It's being decreased over time. And that's the fundamental problem of all this. So, you know, it's a question of how fast are these cycles going in your field from year to year? How much is being decreased? And again, you know, if you're monitoring, you know, how much respiration's coming out, you're doing something like that, you know, that was why those systems that Dr. LaSalle was pointing out where he was measuring CO2 production off of different fields. That's what he's trying to do. He's trying to figure out what really is changing in the system. Because as I said, one of the ways to measure the decomposition of the microbial activity is to see how much carbon is being respired off. So those infrared sensors that detect the pulses of CO2 are crucial for all that. And of course, and with this system, all you're going to do is respire all this off the CO2. So that sort of hopefully ties some of this together. This whole question of carbon size and quality at the chemistry level is still part of the ongoing discussion about really what those compounds are. The basidiomycetes and the ascomycetes drive the decomposition. That we know. That's not questionable. It's a question of how all that fits in to what are plant-derived and what are microbial-derived. Okay, let me see how are we doing for time. Okay. Any questions about it? Because we're almost at the time for Q&A for this afternoon. Everybody sort of burned out already? Yes?
Okay, so I've heard you speak before about, carbon in the atmosphere versus the carbon we have in our soils and how that's changed, pretty drastically here recently. What has caused the excess carbon in the atmosphere, and how is that actually affecting these processes in the soil?
So that's a good question. Without humans, the carbon that goes back and forth in the atmosphere is part of that cycle where the organic carbon is being converted to its inorganic form by decomposition and mineralization. So when you breathe in and out, and you breathe out CO2, you're mineralizing the pizza you just ate, all the organic carbon in it, through our cellular processes. All life does that. All the bacteria, all the fungi convert organic carbon to its inorganic form. That CO2 goes up in the atmosphere, and what takes it out of the atmosphere are all the primary producers that carry out photosynthesis. And so they're fixing that carbon into plant material. Or if they're cyanobacteria, they're fixing it into green algae, right? Now, the problem that comes, now, that's one part. The other part is that, like any organism, how is your mineralization of carbon when you're sitting versus a mineralization of carbon when you're running and jogging? Which is more? Running and jogging, right? Because you need the ATP. So whenever we change our active state, we require more energy to do that. And as a consequence of that, we put more CO2 out of us. Well, the same thing's true of microbes. When it's warm and it's wet and there's a lot of carbon and enough nitrogen, they're going to build biomass and they're going to respire more carbon up. So there's this normal pattern of CO2 in the atmosphere and being taken up. And again, that occurs whether humans are here or not. Now, when humans are back in the equation, what do? Well, in agriculture, what do? If we till, right, what we end up doing is breaking up mycelial networks. They have to repair themselves, so the respiratory activity has to go up. Because what are they doing? They're trying to fix themselves. And so the respiration rates out of soil that's been disturbed has always been higher than soil that's not. Now, and again, that's not a static parameter. If there's organic matter that comes in, I mean, if a mule deer walks across something and poops in a field, that extra carbon is going to be used, and the respiratory rate of that little area is going to go up. Has to, because they're using that carbon. But if you're doing 2,000 acres, the respiratory activity of the landscape is going to go up tremendously, because you're respiring all that carbon that's in those 2,000 acres. The microbes, the fungi, and the bacteria are repairing themselves. And the bacteria, particularly if there's nitrogen. Again, this is all related. If there's no nitrogen, they're not going to do much. They're simply going to sit there and not respire. But if you get them nitrogen, they're going to respire. And then we come along with burning fossil fuels, and we put even more CO2 up. Because what are we doing? We're just mineralizing carbon. That's all. That's what an internal combustion engine does. You burn anything, you're mineralizing the organic carbon, taking the calories that are in it out as heat and energy, and you're changing it to CO2. And it's going to go back up in the atmosphere. And that's why our CO2 levels have gone up. Because we're doing it more than what the planet's doing naturally. Does that answer the question? Yeah, so
that, I guess my next question, that does, but the follow-up to that is the excess carbon in the atmosphere. Is that a lower amount of carbon in our soils? I mean, is there a fixed amount of carbon in our world here, in our Earth atmosphere?
no, because the majority of carbon in the atmosphere is predicated how much is stored in the biosphere. So what we're doing is we're releasing all the stored carbon in the biosphere through burning fossil fuels that has not been up in the atmosphere for eons of time. And again, part of it is this. When that carbon was in the atmosphere, there were more plants to pull it in, right? And things just worked at a different level. The problem we've gotten ourselves into is that we're releasing more carbon, whether we do it through destruction of land or burning of fossil fuels. That CO2 has to go someplace. Now, what's the other reservoir of CO2 on the planet besides the atmospheres? It's the ocean. It goes into the ocean. But what happens when you keep putting CO2 into water? You make beer, right? right? You carbonate stuff. And so what's happening is we're acidifying our oceans. We're making carbonic acid. And so part of the whole notion is, this whole issue of carbon sequestration. For us right now, as a fungal ecologist, we have two options dealing with the climate issue due to CO2. We need to stop putting it up in the atmosphere. We also have to increase the amount of carbon that gets reabsorbed, not in the ocean, but on land. And the best way to do it right now is to put it through plants into soil. And it goes back to all the issue about carbon storage. Because you can't you ecologically can't do it any other way.
So I'd like, to your question, matter can't be created or destroyed, so it's just in place. But a lot of you know me very well. You know that I like to read every conspiracy theory, and I'm very conservative. I also deeply care about ecology, and I don't think that we should try to fit ourselves in a tiny box. But I hear things that are insanely wrong. I mean, Dr. Zach, I was the biggest skeptic of when people start talking about climate change and this and that. The politics that surround it are awful. But these processes make complete and total sense. And we're so wasteful as a society. We grow 14 billion bushels of corn a year. And we've got it running out our ears, and we feed 95% of it to animals that we can find. Why do we do that? We do it because it's easy, not because it's less money. It costs less to put a pound of gain on them on grass than it does in feed. I mean, we could finish those animals for a tiny amount of time, a fraction of the amount of time on grain. Why don't we? Because if we did, we'd have corn running out our ears that we probably don't need to be growing. We need somewhere to go. Right. We monetize resources. We haven't figured out a way to value resources for what they're worth or what the ecological cost is of doing things the way that we do them. One time, I got to eat with Dr. Zach, and we were in his home, and he said that if we were going to go forward and fix this deal, we were going to have to find another metric to judge the economy by and kind of operate the economy other than consumer spending and consumerism, because it's just inherently wasteful. And I love this day because I'll bet you that there are people in this room that came in here that thought that all of these things that you're patiently explaining to us Cretans were just a total, conspiracy theory. And, you were doing a phenomenal job explaining some very difficult concepts to some people that really, really are struggling and trying hard to grasp them, and people that will share your message and what you're teaching us with others.
I appreciate that. But I think one of the all things that have come out of this for all of us is science is always progressing and reevaluating. So we're talking about this issue of humic acids and really what is humus and whatnot. Right, we really don't have that all pegged on either. We know what the goalposts are. We know how some of the larger things operate. We don't know exactly why it works the way it does yet. We don't know that. All we know is you need lignocellulose and you need fungal breakdown products.
Ariane, I have something that goes with you. I come from more of a cow background, not farming background, so I get it. Bank is knocking on the door if you don't get, good cow prices and whatever. But I've had someone ask me, how do we start putting a price tag or a monetary value to the ecosystem services that things that we're talking about today? How do we start bringing that into the equation of the economics of did we make a profit or not? So when does the ecosystem service become going into profit?
thought.
Yeah.
I don't know what the solution to that is.
Right.
But
I never thought it could be done until 2 or 3 days ago. And Tim LaSalle, changed my opinion on that. I thought that and I still think it's going to be really, really dangerous. And it's going to be hard because there is so much greed and so much corruption, and there are so many bad people, trying to monetize this whole ecosystem services.
Yep.
But there are people on the other side of it, like Dr. Zach and Dr. LaSalle, that are trying to figure out how to do that, how to make it real, to where maybe instead of growing corn that we don't need, maybe we're growing covers that we do need, and we're actually getting paid. And there's not all these people in the middle gobbling up. I think at worst there's a chance that if certain people get their way, that they'll gobble up 97 cents out of every dollar.
Yeah.
And if you don't believe me, look around at some of these charitable foundations that, literally spend 97% of the money they collect in administrative costs.
Yep.
That's, 97%.
Yeah. Yeah. That really is a challenge. Someone had a question back here.
Well, I just wanted to ask that what you're saying, Arlen, is that what you're talking about is ESG, these environmental governance, these larger corporations, it's being regulated. Even if it doesn't come down to legislation and trying to not pass legislation and regulate that, and that companies have to find these credits somehow. And I keep saying, it seems like to me it's another market that's developing for us. If we can figure out how to, like what you're saying, take out the people in the middle and work directly, from one end of the value chain to the other end of the value chain, moving those closer together.
Yep.
So that ESG credit is actually legitimate.
Yeah, but I'm out. That's somebody smarter than me is going to have to work on that. I'm just going to work on something else.
I think we had another question back here.
I went criticizing from the sidelines.
Right,
Yep.
Just for example, there was a peanut company down south that had a European company want to do something similar, but their requirement was they wanted the company down south to unionize their employees. And then they just turned them down and walked away from it.
Mm-hmm.
But they could have offered another solution, like, this would be on that consciousness part, maybe not ecological, but to say, if you'll put in a medical clinic for free for our employees, we'll manage it.
Mm-hmm.
But we aren't going to help our employees unionize. So there's companies that are open to those things. It's just a matter of them finding us or us finding them.
That's just like this old carbon that was brought up, it's almost like another stock market.
Right.
Because there's no standardization on how we're going to measure this.
Right, that's right.
Here, a lot of, they've been doing this no-till 10, 15 years. Are you going to get credit for that? Because some of these companies are now telling you you've got to add something else to get onto their program. And there's no standardization. So who's really making money?
Right. Hold on, we have one gentleman back here.
In your chart there, you were showing the contribution to soil carbon coming from plant parts being the primary contributor. Does that include the soluble carbon via the revexinates? Is that included in that?
Yeah, that's all. That would be all part. But the problem with the soluble carbon is what's running the microbes on a short-term basis, but it's not contributing to the long-term stuff. That's the more recalcitrant carbon. That's the lignocellulosic material. So you need both. You can't have one. So if I went out and if I put out basically, and this is what happens, as you well know, if you put wood shavings on your garden at home, what happens to your garden? It slows down and stops. It's the worst thing you want to do, right? Because the lignocellulosic material is so high in carbon and nitrogen, it's going to pull nitrogen out of your ground, and everything's not going to grow well, right? Until that ratio gets balanced again, and then you get. So the thing is, for all ecological systems, you need both to be able to function, because you need the microbes to be able to quickly return things to the plant over the growing season. The other part is the decomposition is the long-term component of the actual dead plant material. And so you have to, and so in these, see the problem with cotton is in a normal cotton field, how much carbon above ground goes in over the growing season from above ground parts? Almost none, except for what's coming out of the roots. So your roots during the growing season are feeding what the plant needs. But the plant as a perennial is expecting to be there for 20 years. And so it's dropping leaves and doing things to build soil carbon as part of the ecological system, not for that year, but for 20 years period. And that's a much different, and see, because we're growing them as annuals, though we know they're all perennials, and so the system is geared differently to how these things function. And so the plant and the microbes are trying to do both. But the problem is we're only managing it from a year to year to year basis. That's the difficulty. And we don't take the long term. So to compensate for that, that's why you need the stubble. Because you got to put something else on there to compensate for the lack of having this input in on enough period of time for the system to run over that 20-year period. And that's the long term. You're not going to build anything up. I mean, if all you grew were annuals, as we mostly do on a field, you don't get enough long-term carbon storage on it. I mean, you will, right? You will, but you got to have enough stuff that remains to be able to do that from year to year. And right now, I would tell you, we don't know what that threshold is, how much you need. I mean, and that may be why we don't get a lot of increase. But then, okay, what happens if I have 8 inches of stubble on the ground? Can you plant into that? No. It's a balance of what that means.
Well, I guess that's what I was going to maybe add is if you look at the world, within the last 200 years, you had natural, fairly undisturbed ecosystems across the entire United States and South America, North America, South America. Then we came in, we cut down all the trees, we oxidized all the carbon up in these grasslands. I mean, that's got to make a huge contribution to CO2.
Right.
And it also, they were functioning without fertilizers.
Right.
And I guess one of my questions I have is, so if we want to build productivity back into our soils and actually build topsoil, we've got to add carbon, which means we've got to be photosynthesizing, and it's mainly going to come from grasslands.
Right.
Just legumes. Legumes, I think, get that nitrogen credit because the carbon to nitrogen ratio is so narrow. So if I'm trying to grow all this biomass, but yet I'm also trying to grow crops, and it's screwing up all this cycling, which I'll admit, as I get further down this rabbit hole, I'm starting to think, why am I growing crops at all? And bringing livestock back and planting perennials. But until I get there, I can capture carbon through photosynthesis, but don't I kind of almost have to add nitrogen to a certain degree to not collapse the crop I'm trying to grow?
No, you do. And so part of it is you have to understand, again, at the current state you're at, it goes back to my original question. It's not the pool of nitrogen that's there at any given time. It's the rate at which mineralization of that organic nitrogen comes back to inorganic form to feed your crop and to feed the microbes. We don't know that because we don't measure it. We don't measure that. The other part is, I mean, the only way for nitrogen to come out of the atmosphere naturally into the soil is through legumes. In fact, I don't know any terrestrial system that doesn't have legumes in it. Right? And now, that's not just legumes. It was mentioned, I think Dr. LaSalle mentioned it too. There's a whole group of bacteria that are free living that also fix nitrogen, right? That are on plant leaves, on plant roots, that don't have any symbiotic association. But we don't ever think about those. And they may not add a lot of nitrogen, but they add nitrogen, and they're part of the normal way ecological systems function. But the problem for us is we want the same response year to year to year to year, And we're in a step function where we're doing little increments of change. And so the question is, because you're not going to change it completely in a couple of years, the idea is to figure out how much the system can recycle under a given set of conditions, not just carbon, but the organic carbon with nitrogen to an inorganic form. And if that's high enough, you shouldn't have to be able to do anything with it.
Well, yeah, because the point of growing organic matter is to build a bigger nutrient and water cycle that flows through. It doesn't just stay static.
That's right, right.
It's always cycling.
It's always cycling.
So if you can, and a lot of times in ag, we just talk about everything, in order to have a framework, we talk about the 6 inches.
Right.
But, so we can get that to another foot or down to 2 foot, and you think about all the water.
Yes.
That's where I'm starting at a degraded state, and I'm trying to build this and not, I guess, make the crop nutrient deficient.
No, and we were talking about this this morning too. And here's the problem too. The stubble and material from the previous year is not at the state at any given time for you to have enough nitrogen necessarily for the spring, because there's just not enough time. Right, it needs the microbes, because again, as it goes over winter, the microbes need some time to be able to so you're going to have to add some nitrogen to sort of work the system through. But once you get to a certain stage, again, it goes back to mineralization. If we knew how much mineralization, how much nitrogen needed to move through the system per unit time to grow 2 bales an acre, that's the number you want. Who cares about what the static pool is? You want to know how much nitrogen the system, through deposition and through the fixation of nitrogen from the previous year, is now being made available. And once you know that, then you can plan for whether you need to fertilize or not. But we don't do that.
Yeah, that's what he and I were talking about this morning, is that you can use a cover crop in the winter to keep that nitrogen from leaching in the wintertime, or being lost to the air. That plant will take it up, but then we don't have enough information here in this region on when's the right time to terminate that cover crop and use it as a green manure, so that we can get that nutrient cycle back in because of our variable climate.
Right.
So, if you do it too soon, then, or you're doing it too late, then it's immobilized or it's not broken down yet. So then it's not ready when the cotton needs it. Or if you do it too soon, then your microbes have used it and you may lose it to leaching during the spring rain. So that's what I was talking to Dr. Zach about, is that we need a little more of that, like looking at what's the timing of these cover crops in this region, which are the right ones to use that would match the water and the climate.
Well, that's one of the things I guess I'm struggling with, and why I'm kind of asking the question is, so we plant rye. If we're not planting wheat in the fall, we're going to plant a rye. And, one of the things, if I'm going to plant another grass into a grass, like corn, I want to terminate the way I'm trying to manage it, whether it's right or wrong, is I'm trying to terminate it while I'd say it's in the vegetative. So it's like a 30-to-1 carbon nitrogen ratio. And then in order to make sure, just as a guarantee, to make sure that I'm not inefficient, I'll go put on 10 gallons of 28% in order to kind of cycle that quicker, right or wrong. I don't really want it to go what I would call reproductive, or it starts to make straw, gets the lid.
Right, yeah.
Because then my carbon to nitrogen ratio goes 80-to-1, and I'm going to have to add a lot. And I don't know and it just gets really tricky. Now, planting beans into that, I don't have any issue.
Right.
So I guess that's where I'm trying to go. I'm not maybe asking you the question.
No, and it's a good question.
I struggle with I know there's all these different carbon to nitrogen ratios, and then I hadn't even really thought about how the biology, or maybe trying to rob carbon. I always thought, okay, well, the biology is going to take, the way they'll starve it is they'll take all the free nitrogen, my understanding, is out of the soil to try and start to break down all that residue. Again,
knowing how normal systems function, your stubble coming from the winter to the next growing season is not for that crop. It's for the crop a year ahead. It takes at least that time to get everything in any given time. And it's doing the same thing, because don't forget, in a natural system, those leaves are dropping, that material sits there, but that's at the top of the pile. What's going to provide the nutrients for the current growing season is what's at the bottom, but that's been deposited 2 or 3 years ago. That's the difference. We're working on a year-to-year time frame, but the biology is not a year-to-year event. You have to produce it, but you're actually feeding the crop a year to 2 years ahead and getting that system in gear to be able to do that. That's why you need to know what the mineralization rates are before you plant and during the planting. I can't stress that. We need to know how much nitrogen is moving before you plant to know what the capacity is of that soil, not from what you did this year, but what you did 2 years ago, because that's what's generating that material. Because it just simply takes that amount of time to do it.
And you talked about that CO2 monitoring.
Yeah, so you can measure CO2 as a way to know what the respiratory activity is of the microbes. That would be one. But if you're really crucially interested in nitrogen, we need to know the mineralization rates. We need to know how much is moving per unit time. And we need to look at what our plant demand is per unit time. Because again, ecological systems are not based on a slug of nitrogen. They're based on continuous nitrogen.
So if we knew that mineralization rate and we needed to add a slug of nitrogen to make our system work.
Yeah.
In my mind, the time to do that would be the coldest point of the year.
No,
you have the minimum impact on your microbes?
No, you want to look at, because again, your fungi and your bacteria are going to be active as temperatures start to warm up and work on the carbon. The question is, you don't want to put too much nitrogen on, because all they're going to do is decompose. Right? They're simply going to use that carbon. What you have to balance out is how much decomposition do you need per unit time, and the way to measure that is the amount of nitrogen moving through the system, because that's linked up with the bacteria and the fungi. And that's really going to give you the key. And again, we have to know, and you should be able to do it, per unit time, how much nitrogen do cotton plants need to take up.
Okay, so maybe I'm looking at it completely backwards of where I should be. I should be looking at the mineralization rate and then looking at my crop and saying, I should be looking at my yield goal and what I'm trying to achieve based off of my mineralization rate, not the other way around.
That's right, yeah. And then if your mineralization rate won't provide the capacity to get that, then you have to fertilize.
Thank you.
Yeah. Yes, sir.
Steal Dr. Zach's brain.
That's okay.
When you're talking about measuring that mineralization rate, how are you following that?
So I was mentioning earlier, there's a couple companies out, and we've used them. They're an ion resin exchange stick, and one's for nitrate and one's for ammonia. You simply activate them. They have nothing on them. You activate them, stick them in the ground at a certain depth. You pull them out and you send to the company. And what they do is they say, okay, this is how much nitrate or how much ammonia is on there, and you leave them in for certain amounts of time. So I can tell you per day how much nitrogen is moving through the system. And by that, I can calculate. Now, if you really are interested, what I can do is I can put a stick in on one Monday, put another stick on the following Monday, because don't forget, these are cumulative of measurements. So all the nitrogen and ammonia could come in one day, right? And nothing rest. So what you also have to do is sort of balance it out to see what's the real time frame. But it depends upon the scale. If you want to leave them in for a week, you leave them in for a week and say, okay, in my field, what's going on the first week of April? What's going on the second week of April? Third week of April? Fourth week? We don't have enough information to actually pin it down like that. I mean, we measure mineralization, and we simply look at, okay, what's the capacity of the system to actually convert organic forms to the inorganic forms that plants can use. Because if they're moving to the plants, they're moving to the microbes. We don't worry about that, right? We worry about what's plant available nitrogen, and that's where these are going to tell you. Because now, if after doing all this, your mineralization rates are really low, even with adequate moisture, then it's all locked up. There's not enough nitrogen in the system. And then you have to do something else. Yes, sir.
What about the play of your micronutrients, especially if you've got some severe deficiencies?
Yeah, I mean, my take is add them, right? Because there's going to be some things that the microbes are going to be able to do, but there's not going to. They do. The mycorrhizae do move. They move nickel. They'll move iron. They'll move calcium. They'll move it around. It just depends upon how severe it is.
I hear talking to other fertilizer reps and stuff. I guess zinc and boron.
Right. Right.
Big ones,
Yeah, there's not a lot that the microbes can do for boron. It's just the nature of what it is. Yeah. You have to. Yeah.
You're talking about those strips. Is that the same thing as a microbiometer?
I don't think so. Right. I'm trying to think of the company. I'm trying to think of the company in Canada that used to do them. But you should be able to see them online. Interestingly enough, if you knew someone that ran soil tests, you can build them. We used to build our own. You can buy ion exchange beads. You put them in. We used to buy pantyhose, wrap them up in pantyhose, activate them, bury them, and then collect them and extract them. We could do it ourselves, and then you have to get somebody to measure it. But they're easy to do. But the company already sells them pre-made, and all you do is activate them and bury them.
Kind of back to Eric's question a little bit. Currently with soil fertility, you look at what your crop grew off, what it could potentially put back, what your yield goal is, what your water level is for the next year, and then you have basic guidelines on how much NP that you need.
That's right.
And so you make a proactive decision, to apply so much nutrient.
Mm-hmm.
So, we were talking about these sticks you put in, and you get your mineralization rate. Can you assume that if you have a very high mineralization rate early, that's going to continue all the way into flowering? Because once you get past squaring in cotton,
you're not going to be right.
So can you make that assumption that if you have high mineralization rates prior to squaring, there's going to continue on into bowl opening or?
No, because we don't collect that kind of data that much, right? So that's something that has to be done. We don't collect I'm sure there's mineralization rates for soils in there somewhere, but it's not used effectively in the agricultural literature. There are projects and experiments all over the country on mineralization in controlled situations where they know every month how much moves through the system, right? But and I'm sure some of the long-term agricultural ones have some information on that.
So are we talking parts per million per day as a mineralization rate? Is that the data that you get back?
Yeah, that's exactly right.
So how many parts per million per day does it take cotton for 2 bales?
So what you have to convert is that parts per million to whatever the weight is of nitrogen you need to add over the growing season. And can you get that simply by mineralization alone? That's what you have to figure out. Yeah.
Dr. Zach, back to his question about the micronutrient.
Right.
If you've got a shortage there, that's going to affect your microbial community as well.
It will.
Those are coenzymes.
Right. Yep.
So is it safe assumption that it's not just the plant maybe suffering just from that micronutrient, but from the inactivity maybe of some of these microbial activities that could be missing?
It's possible, but I'm not familiar with anything that mineralizes boron.
No, right.
Right. Now iron and manganese. Fungi. Interestingly enough, the basidiomycetes are major elemental mineralization components of calcium. They do a lot of stuff with calcium because for fungi, calcium is important. And so the basidiomycetes actually change calcium levels a lot where you have them. And they do that for various reasons. But beyond that, now bacteria and fungi need iron, So, but that's all based on pH. So a lot of this is abiotic aspects. And then also what your cation exchange capacity is in your soils relative to those nutrients. And that's going to be affected by your soil organic matter. So that's how it all interplays.
Yeah, I know one of the guys we talked with, that cation exchange, you can look at that number, I can't remember what the multiplication was, like 10 times or something, with all the nitrogen that that plant or whatever.
Yeah, that's right.
Take it one time.
Right, yep, yeah. Yeah, so you sort of have to take, because soils are not doing one thing at a time, they're doing all these things simultaneously, and they all affect each other in some way, shape, or form. So the amount of sodium and calcium, the amount of clay particles and soil organic matter particles, what size pores you have in your soil, all that's going to interplay as to what these systems are capable of doing. But in a gross scale, and again, I think it goes back to what can you really manage? And in soils, the thing that you can manage is organic matter content. And if you can manage that, everything else starts to fall into place. That's the major thing. That's why it's always about soil organic matter. Because then everything else will because the plants as they decompose are going to put the micronutrients back in. It's just a question of where they are and what they're bound up to. But that's all organic matter driven.